Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Michael Lebrecht from Repligen.
Thank you, Leah. So today our discussion will be about the impact of Repligen CS single-use TFF products in downstream processing. CS TFF cassettes are well suited for concentration, diafiltration, and buffer exchange in downstream processing. They're offered in a purpose-built, single-use, flat-sheet cassette format. Our unique manufacturing process provides a TFF cassette which is more user-friendly and more economical for use in single-use applications, particularly in cGMP and clinical manufacturing scales. Our CS single-use TFF cassettes are pre-sanitized and delivered in 0.2 molar sodium hydroxide, and so they're ready to use right out of the box. They can be installed into conventional TFF holders from all the major suppliers, as well as Repligen TFF cassette holders that have a fully disposable flow path. And we'll talk a little bit more about that later in the presentation. Our discussion today will also describe how the CS single-use TFF cassettes will save time and money, reducing filter costs by 80%, reducing labor costs by 50%, and reducing buffer costs by 75%. But first, I'd like to describe the two membrane chemistries we have available. Our TFF membranes are specifically formulated for narrow pore size distribution and efficient selectivity, providing reliable and repeatable membrane performance, as well as delivering fast permeate flux and high product recoveries. Both our membrane types are based on modified polyether sulfone. Our ProStream membrane is available in a range of molecular cutoffs from 1 kilodalton to 300 kilodalton. It's a neutral charged, low protein binding PES and has similar characteristics typical to other PES membranes on the market today. Our HighStream membrane is more unique in that it's more hydrophilic than other PES membranes. It's specifically been modified to be hydrophilic and appropriate for applications where cellulose membranes are typically used, particularly with applications for monoclonal antibodies. The HighStream membrane is available in ultrafiltration cutoffs from 5 kilodalton to 300 kilodalton, and in addition we have some microfiltration range from 0.1 micron to 0.65 micron. Our CS and CSPD cassettes are available in a wide range of surface areas. The CSPD cassettes are available in 0.01 meters squared or 100 square centimeters, 0.02 meters squared or 200 square centimeters, or 0.1 meters squared. These PD cassettes are well suited for evaluation, screening, and process development, and are directly scalable to the larger surface area cassettes used for clinical manufacture, pilot plant, and even production scale. The CS-TFF cassettes are available in larger surface areas of 0.5 meters squared, 1.5 meters squared, and 2.5 meters squared. These cassettes can be stacked on top of each other to create larger surface areas, and they can be installed into traditional format TFF holders from all the major suppliers. And again, they can also be installed into the Repligen format, which has a fully disposable flow path. So now we can take a closer look at the impact of single-use TFF format in downstream processing, and we can consider the cost of traditional reusable TFF cassettes compared to the CS single-use TFF cassettes. So we did a survey of the major suppliers of reusable TFF cassettes in the market today. Right now, the typical price for a reusable TFF cassette is about $5,000 per meter squared. Because of our unique manufacturing process, we're able to sell our CS single-use TFF cassettes at 1/5 the price, or 80% of the price of a reusable TFF cassette. It's just at under $1,000 per meter squared. Additional cost savings can be realized by reducing labor that's typically found in most reusable TFF processes. So if we take a look at the graph here, at the bottom, we have a representation of a typical reusable process. And typical of most reusable process for TFF is going to be 10 steps. Typically, you're going to have a reusable TFF cassette is going to contain some type of a storage agent, typically glycerin, or some type of a bactericide. And so that storage agent is going to need to be removed before use. So in the reusable process, we start with installation of the cassette, integrity test, and then we have to do a pre-use sanitization to remove the storage agent. From there, we flush the cleaning solution out and have to perform a water permeability test. And typically, you're going to want to do a pre-use water permeability test to establish a baseline of water flow rate in the new cassette, because if you're going to reuse the cassette, you need to measure the water flow rate before and after to make sure that your cleaning efficiency is correct. From there, we get to the equilibration step, and then finally to the actual UFDF process. In the reusable process, again, we've got to, at the end of the UFDF process, we're going to have to clean the membrane, sanitize with a sodium hydroxide, flush that out, and then repeat the water permeability test, again, to establish whether the cleaning efficiency was adequate. And from there, we can go to storage of the system. So in this case, let's say the actual UFDF process was taking 6 hours. There's about 3 hours of time at the beginning, about 3 hours of time at the end to do the cleaning and the flushing and all these other steps to get the cassettes ready for use or post-use. So all told, you're talking about a 12-hour process. And typical of TFF steps, TFF days typically in clinical manufacturing. It's typically an early morning start and a late evening finish. It can be somewhat unpredictable in terms of the runtime for a reusable process. We compare that to the single-use process, where now we've reduced the number of steps from 10 down to 3. In the single-use process, now we're doing an installation, an integrity test, and we're going right to the equilibration step because these cassettes were pre-sanitized and delivered in 0.2 molar sodium hydroxide. So we just need to do equilibration to neutralize the pH, and we can start the process. So that same 6-hour UFDF time, we're only talking an hour or two for pre-installation and prep time. So the single-use process, under 8 hours total. So much more useful to operate in a single shift with the single-use approach. So reducing the labor and the time for processing can increase facility throughput and productivity. Another area of savings is reduction in buffer usage. So if we think of that previous slide where I showed you the 10-step reusable process, we can assign a volume for each of those flushing and cleaning steps. And if we were to totalize each of those volumes in terms of liters per meter squared membrane surface area installed, that would totalize up to be about 230 liters per meter squared in the reusable process. By eliminating the flushing and cleaning steps in the reusable process, we can reduce that volume to 60 liters per meter squared, or a 75% reduction in buffer usage. And we can also consider the cost savings in regards to the QC costs, transportation costs, and other costs for making the buffers and testing the buffers as well. Our CS-TFF cassettes are fully scalable from process development through clinical manufacturing and into production scale. And they can be run at transmembrane pressure and cross-flow rates that are very typical of what you'd use in a reusable TFF process. They are linearly scalable from the 100 ml scale all the way to the 2,000 liter scale. And we have installations, end users that have installations up to 30 meters squared and greater in our single-use format. We also offer our CS-TFF holders with fully disposable flow paths. Here we're using a USB Class 6 polypropylene manifold to connect tubing and disposable pressure transducers and other single-use instrumentation we're connecting directly to this, the tri-clamp connections on the manifold of feed, retentate, and permeate. So in this case, there's no fluid contact to the stainless steel end plates, and we completely eliminate the need for cleaning of the TFF holder. And this holder format is available in benchtop scale all the way up to production scale. Repligen also offers a urethane encapsulated reusable format with our Pro Series TFF cassettes. These are more traditionally built cassettes, and they're well suited for repeated CIP and reuse cycles. They're also available in the same surface areas as our single-use, as our CS single-use cassettes. So the same surface areas, low process development scale and process scale. So important point here is as membrane area increases with, say, late phase clinical or even commercial scale, the process scale economics for TFF can swing back in favor of reusable format. So our Pro Series reusable cassettes are directly scalable from the CS single-use TFF cassettes. In fact, the exact same membranes available for the single-use and the reusable formats. So for example, if you were to begin using our 30K high-stream membrane in our single-use format in a clinical manufacturing scale, you can get that same exact 30K high-stream membrane in our reusable format if your process economics favor reusable when you get to larger scales. So here we see an example where showing where the membrane area does have a big impact on the process economics of whether you're going single-use or reusable. The graph on the left here showing a smaller installed membrane surface area of 2.5 meters squared might be typical of a clinical manufacturing batch. But here at this scale, the factors of labor and buffer savings have more significant impact on the cost per run. So the single-use cassettes have a more favorable cost per run compared to the reusable TFF cassettes. With larger installations, looking at the graph to the right here, showing an installation of up to 40 meters squared, so it might be more similar to commercial scale manufacturing. With larger installed membrane surface areas, the cost of membranes has a more significant impact and influences the cost per run greatly. So here we're looking at the reusable TFF cassettes having a better cost per run or more favorable cost per run. So typically we find that the time savings and the increased facility throughput realized by the single-use TFF process is particularly well suited for contract manufacturing operations. Our single-use and reusable TFF cassettes are fully supported with our validation guide, which includes information on leachables, extractables, and other product safety information such as USP Class 6. Each of our TFF cassettes are individually integrity tested and provided with a C of A that indicates their compliance with our quality release testing. So in summary, our Repligen CS single-use TFF cassettes have a highly positive impact on cost savings and increased productivity in downstream processing steps of concentration, diafiltration, and buffer exchange. Filter costs are reduced by 80%, labor savings by 50%, and buffer usage reduced by 75%. So hopefully Repligen can bring that same increase in productivity to your UFDF processes. And with that, I thank you for your time, and we're available for questions.
Okay, thank you, Michael. So we do have some questions. Do you have a similar technology for spiral round crossflow system?
The Tangentix format is only available in flat sheet cassette format. We are compatible with cassette holders from all the major suppliers of TFF cassette holders, but it's only available in flat sheet format.
Okay, so the next question is for reusable cassettes, why would you need to run a pre and post normalized water permeability test?
For reusable? Well,
yes.
You need to do a pre-use water flux test to establish the baseline. Typically that's something you're going to run when the cassette is new, the first use. You're going need to establish a baseline clean water flux on that particular cassette. Suppliers can give you some water flux information, but you really need to test the actual filter or filter installation on the water flux. So you only need to do that pre-test on the first time, and then when you are reusing a cassette and cleaning it, you need to do the post-use water flux to compare the recovered water flux to that original value. And you do a ratio. Typically the guideline is to have at least 80% recovery of your water flux to establish that the membrane cleaning has been efficient.
So we have another question. We currently use 2200 centimeter 500 kV TDFF columns. Do you have something comparable?
Yes. That would be, that's roughly about 0.2 meters squared. So we have 0.1 meter squared cassettes. We could stack two of those on top of each other to come close to that, very close to that surface area. So so that's what's good about the cassette format is you can stack cassettes of different surface areas to create really any surface area that you might need. So yeah, we could meet that requirement.
What type of molecules would be better for the high stream, and which proteins are good for the pro-stream type membrane?
Yep. Well, the significant difference between the two membranes is that the high stream membrane is more hydrophilic, and it tends to have performance that's going to be similar to other cellulose membranes that are in the market, and particularly those hydrophilic membranes are very appropriate for use in monoclonal antibody applications. When you start looking at other recombinant proteins, really you can look at both membranes, and often we'll do a side-by-side comparison of our ProStream membrane, which is a neutral charge, low protein binding membrane, and we'll do a side-by-side evaluation with our high-stream membrane, which is the hydrophilic, and really let the testing establish which is the best membrane choice for the given application. We've got field application support that can come into your lab, and help you do those experiments to establish which membrane is better performing on your molecule. And we have very low holdup volume systems. Our small surface area cassettes only have about a 2 ml holdup volume. So with just 100 mLs of sample, we can do a pretty thorough evaluation on membrane compatibility and screening.
Okay. And another question. We have quite a few questions. I'm not sure we'll get to all of them. If we don't get to them in this Q&A, they will be given to Michael, and he can follow up with you directly. So another question, will higher MWCO membranes be available in the future?
Right now, the range that we have presented here is our validated range. We go up to 300 kilodalton in ultrafiltration, and then we go to microfiltration. So we really don't have much room in between. I guess there's a 750 kilodalton that you could see available from, say, some of the hollow fibers out there. One of the things, if we really look specifically at our selectivities, we want to have unique selectivities from each membrane. We don't want to have any overlap of specification between one cutoff and the next cutoff. So it kind of makes it difficult to have other cutoffs between the 300 kilodalton and the 0.1 micron. So at this point, the offering we have right now would be what you could expect in the future. But we could take a closer look at what an individual process requirement is and what cutoff that we offer could be appropriate for their application.
Okay, I think this will be the last question. Can you sterilize the cassette by gamma irradiation or autoclave sterilization for use in an aseptic process?
Okay, that's a great question. The answer is no, unfortunately, at this time. This product is pre-sanitized in 0.2 molar cyanide dioxide. It's delivered chemically sanitized, but it is not compatible with gamma radiation, so it's not appropriate for aseptic or sterile applications. Typically, you are installing these cassettes into a cassette holder, so it is an open system. You're assembling the cassettes, you're putting the holder top on, and then torquing it. So it's not a closed system, and it's not an aseptic system in the offering that we're talking about here today. But it is chemically sanitized and ready to use out of the box. Okay.
Okay. Well, thank you, Michael, and thanks to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess Internationally Ask the Expert webcasts covering many aspects of bioprocessing. Look for those announcements in your inbox. Thanks for attending the webcast today, and have a great day. Goodbye.